In this video, we will learn and understand how to use counting chamber or hemocytometer or neubauer's chamber in a highly easy and logical way. Please watch the video till the end, and I assure you that you will understand the logic and math behind it. Your concepts will be 100% clear, and trust me, it's very interesting. You will be amazed by these traditional art of counting millions of cells in just minutes manually with these simple device and technique. Feel free to pause the video whenever you want the detailed description on the screen. In these video, basics of improved neubauer's chamber are discussed. So neubauer's chamber or hemocytometer is used to calculate blood counts like RBC count, WBC count, platelets count, or any cell count involving any body fluid like sperm count or other body fluid leukocyte count. Neubauer's chamber have known and fixed volume with markings, which helps to calculate cell count in unknown sample. So first of all, we will understand what is blood count, like what is RBC count or WBC count or platelet count. So imagine a cube having length, height, and width of 1 millimeter. RBC count simply means how many RBCs are there in a cube of blood having length, height, and width of 1 millimeter. Same as for platelet and WBC count. If someone's RBC count is 5 million per cubic millimeter, then it means there are 5 million red blood cells in a 1 cubic millimeter of blood, and 5 million is not a small number. Volume conversion is also very important, and it will be helpful later in many aspects in medicine. Let's imagine a cube having length, height, and width of 1 centimeter. Then its volume will be 1 cubic centimeter, or 1 cc, and we all know 1 cubic centimeter is equal to 1 milliliter, or 1 ml, that's 1/10,000th of a liter. Now 1 centimeter is equal to 10 millimeter, so 1 cubic centimeter is equal to 10 into 10 into 10 is equal to 1,000 cubic millimeter, that's 1/10,000th of a milliliter. As a general rule, 10 raised to minus 3 is milli, and 10 raised to minus 6 is micro, thus 1 cubic millimeter is equal to 1 microliter, that's 1 millionth of a liter. And it really represents very small volume. So 1 cubic millimeter, 1 microliter, and 10 raised to minus 6 liter, all are just the same. Imagine if we are counting individual RBCS for RBC count. That's in millions of cells per just a microliter of blood, then it might take months to calculate RBC count of a single sample. That is practically impossible. So what can we do? We can do two simple things. First, we can count cells in less volume, then multiply it with appropriate factor, like for example, count in 0.1 microliter volume and multiply it with 10. Second, we can dilute the sample and multiply it with appropriate dilution factor. Because of these two things, our work will be reduced significantly. Now for the cell count using hemocytometer, we require main two things. First, volume of a counting chamber, and second, dilution factor. Now first we will see dimensions of neubauer's chamber. There are a total of 9 equal squares. Each are having length and width of 1 millimeter, so area of one large square is equal to 1 millimeter square. But for the calculation of volume, we also require thickness or depth. How can we have that? Before it, we will see that neubauer's chamber have 2 lateral platforms and 1 central platform, which have 2 counting grids. We place cover slip over central platform and place our sample on central platform using pipette. And sample will be sucked by capillary action. If sample is more, then it will be drained into lateral grooves, which separates central platform from lateral one. If we view from side, then this lateral platform is exactly 0.1 millimeter high than the central platform. That means depth or thickness of a sample film will be exactly 0.1 millimeter. So volume of a large square will be 1 times 1 times 0. 0.1 equals 0.1 cubic millimeter or 0.1 microliter. Volume of a whole 3 by 3 counting grid will be 0.9 microliter. Now, how much volume of fluid we want to evaluate for a count totally depends on ourselves, but there are some general considerations, like if cell count is high, then we need to count less area. As a basic protocol for a WBC count, we calculate cells in 4 large corner squares, and for RBC count, we calculate in 5 small squares in central square. Also in each large corner squares for WBC counting, there are 16 equal small squares. What will be the volume of such small squares? It's very simple. Volume of one small squares will be exactly 16 times smaller than one large square. That is 0.1 over 16 cubic millimeter, or 0.1 over 16 microliter, or equals 0.00625 microliter. At other end, the central RBC counting chamber have also equal 25 squares. Volume of such single square will be 0.1 over 25 cubic millimeter, or 0.004 microliter. If we further zoom in, such each squares are further divided by equal 16 squares. So volume of such little squares will be 0.004 over 16 microliter, or 0.00025 microliter. That's really very small volume. At this level, one might have questioned that why the central square is divided by such very small squares repeatedly. The answer is to provide the user very little volume for counting very high count cells. That makes perfect sense that RBCs are counted in 5 of these central squares. So user can easily calculate RBCs without any confusion. To count one cell mistakenly twice, as user can count such very small volumes, chamber-wise instead of counting in a big square randomly. Suppose if we count n numbers of cells in volume V, then in 1 microliter of a volume, there will be exactly n upon V cells. For example, if we calculate total of n numbers of cells in total 4 large corner squares for WBC count, then volume we counted will be 0.4 microliter, but WBC count is WBC per 1 microliter. So we have to divide that n by 0.4, and thus WBC count will be 1 upon 0.4 equals n times 2.5. But still, we just reduced our work by only a factor of 2.5. So here comes the second part. Dilution. For dilution, we use specific diluting fluid as the blood, or a sample can have different types of cells like RBC, WBC, platelets simultaneously. So specific diluting fluid helps to eliminate unwanted cells. We will see working principles of specific diluting fluids in our latter videos. Now, once we dilute the sample, our work of counting cells will be further reduced. Suppose if we add 1 milliliter of diluting fluid in 1 milliliter of sample, then it will be called 1 gem 1 dilution. Diluting fluid dose not have any cells. So after dilution, total number of cells in mixture will remain constant, but this time in larger volume. For example, if we do 0.5 part sample to 9.5 part, diluting fluid dilution for WBC count, then the dilution will be 1 gem 19, and if first n numbers of cells in the 0.5 volume of sample, then after dilution, n numbers of cells will be there in 9.5 plus 0.5 equals 10 volume. So how can we calculate dilution factor? We can calculate dilution factor by sample volume plus diluting fluid volume upon sample volume. So in this case, dilution factor will be 0.5 plus 9.5 upon 0.5 equals 20. If we calculate n numbers of cells after dilution, then actual count in sample will be n times dilution factor D. So by combining these two things, we can calculate cells in Neubauer's chamber by the formula ND over V. There is also a simple trick for calculating dilution factor. Just convert the sample volume into 1 and adjust diluting fluid volume proportionally. Then if the ratio is 1 gem X, then D will be 1 plus X. For example, if dilution is 1 gem 1, then D is equal to 1 plus 1 is equal to 2. If dilution is 4 gem 2, then it will be 1 gem 0.5 and D will be 1.5. For detailed description, please pause the video and see the table. So finally, we have 3 steps for obtaining count by Neubauer's chamber. First, prepare appropriate dilution by appropriate diluting fluid. Mix it well and calculate dilution factor D. Second, place the cover slip on neubauer's chamber and charge the chamber with above prepared mixture. Wait for 5 minutes. This is really important because the cells will be suspended in mixture. It will take some time to settle down due to gravity, so we can focus them all using microscope on lowermost layer. Third, define how much volume you want to evaluate and calculate the volume V of neubauer's chamber, where U wants to count the cells N. And finally, you will have the cell count by using formula N D over V. I hope you liked the video. In our next video, we will learn RBC and WBC count using hemocytometer. Thank you.